first actual lecture. Today we'll be covering Intro to Science. That'll be both the note sheet and the slideshow. And this, some of what we'll cover today will be on your first quiz and on your first exam. Something I should mention, I didn't mention it in the Welcome to Bio 181, but it's important nonetheless, is that one of your exam scores will be dropped at the end of the semester, and one of your quiz scores, your lecture quiz scores, will be dropped. There will be 5 exams, so there's for each module, so 1 through 4, and then there will be a final exam. So if you are happy with your exam score average after the 4 modules, you don't have to take the final. It will go down as a 0 and then get dropped from your overall score. And then the lecture quiz will be the same. I will not apply those functions until the very last week of school, though. So your exam scores are all going to be kept right now until we're done with module 4, and then I will show you what your overall grade will be once you drop a quiz and an exam. Okay, hopefully that's clear. I just forgot to cover that last time. So anyways, let's get started. I'm going to share my screen. We'll head over to Google Chrome and we will talk about what is science. So we've got our note sheet, Intro to Science note sheet. And we start off by saying when we can't explain something we observe, that's where we're at. All right. But we won't answer that question right away. First, we have to say, well, what is science? What is it, this science thing that we're doing? All right. So let's, in a broad brushstroke, paint what is science. Well, science is it's a study, but it's systematic. That means there's a methodology to it, and it's a study of the structure and behavior of the natural world through observation and experiment. And so what science effectively is trying to do is it's trying to remove the subjective experience of the researchers from the outcome of the experiment. And so what that means is that even if the experimenter believes something, hopefully the way they've designed the experiment will not be influenced, the outcome of that experiment will not be influenced by systems. So it's removing the subjective from our knowledge of the objective. That's the idea of science. And it all starts with an observation. And so an observation simply means that you're looking at something with the intention of gaining information from it. So there's many times where we focus our attention on something, but we're not watching it to gain information from it. So that could be that we're just sitting, looking at the sky, hanging out. But when you see something that you're trying to gain information from, maybe you're driving down the freeway and you see, you're looking around to see what cars are doing. You're observing those so that you gain information about your surrounding and your environment so you can safely travel from one point to the next. And so here we enter this idea that, well, what happens if you're observing things and then you see something that doesn't make sense? What is that? And so we call that a phenomenon. And a phenomenon is just a situation, an observed situation, where the cause or the explanation is in question. So you might see something that begs a question. You might see something that's, doesn't make sense to you. And you naturally, as a human being, start to generate questions about it. Like, what is that? How did that get there? Why is that there? What is its purpose? What is its intention? Is it harmful? Is it hurtful? Is it friendly? So all of these are questions that we naturally ask that proceed a phenomenon, something that you observe that whose cause or explanation is in question. And so, like I said, we ask these questions, we see something, and then we ask, and automatically we ask, well, why did that happen? How did it happen? Can I make it happen again? There's this really funny cartoon I saw when I was in grad school. It was a two panel cartoon, and it says it has a picture of a guy throwing a light switch and then like getting shocked, and then the next panel like, oh geez, I'll never do that again. And then the next panel was like when scientists do this, and so the guy throws the switch and it like shocks him, and he goes, I wonder if that'll happen every time I do it. And so that's kind of what we do. We just say, can I make this phenomenon happen recurrently, given a certain set of variables. And so once we've asked that question, why did that happen? You form this, it's sort of a nebulous hypothesis that's, it's in its infancy stages. And so you see something happen, and you say, okay, why did that happen? And now I'm going to propose a how that happened. So, or you say, how did that happen? And I'll propose how it happened. Or can I make that happen again? Will it happen every time if I do A plus B? Will it always come out with C? And so you can start to form a hypothesis. And a hypothesis is just a working model, a testable working model for how a phenomenon works or manifests itself. So what we like to say, and this is its general definition, is that a hypothesis is simply a testable explanation for that observed phenomenon. So if I see something happen, I ask a question about it, I think I know how it happens, well, then I should be able to explain how it happens and make predictions on what I should expect to see. If I do A and B, then C should follow. And so that's the idea of a hypothesis. And so when we say testable, what do we mean? Well, Karl Popper, one of the famous scientific philosophers of the 20th century, said, if you cannot falsify your hypothesis, then it's not a real hypothesis. So just because somebody comes up with an explanation for how something might work, if you test it and falsify that hypothesis, it's not really a hypothesis. It's just supposition at that point. And so his idea was like, if you can't prove it wrong, then you can't prove it right. And then we would design an experiment. And so a lot of people get confused about the relationship between your testable model for how something works, like your hypothesis, and then a prediction. So hypothesis and prediction often get conflated together, but they're two different things. So a hypothesis basically says, this is my model for how I think this system works that produces this result. Now, when you go to test that model, you design an experiment. And with any experiment, there's always the presupposition that if my hypothesis for how this whole thing works is true, then my experiment should turn out result Z or result Y or result X. So this is the relationship of hypothesis to prediction. Hypothesis helps you design an experiment to test the hypothesis. The experiment will give you a chance to predict the outcome of that experiment if your hypothesis is true. So you might say, okay, I was out in the jungles of Madagascar and I found this plant that was being used by the locals, and they said that it was really good for your heart and it lowers blood pressure. And I say, well, that's interesting. That's a phenomenon. I wonder if we can make that happen. So I have a hypothesis. I say, I think these Amazonians or Madagascarian indigenous people are using this plant to for their health benefits and something along the lines of lowering blood pressure. So I bring the plant back to the United States and I design an experiment to test the model that this plant lowers blood pressure. So my hypothesis is that this plant, if taken, presumably how they prepare it, if you take this plant and you consume it, then it should lower your blood pressure. And so I design an experiment where I'm going to give half the people the plant, half the people a placebo. So they think, and neither one of them knows what they're getting. They just know they're part of a blood pressure experiment. And then what I would do is I would say, okay, I could make a prediction from that experiment and say, if my hypothesis is true, then the group that received the plant medicine should have significantly lower blood pressure on average after it's over than the group that received the placebo. And so my prediction is related to my hypothesis, but they are not the same thing. My prediction is based on my experimental outcome, right? Based on the design of my experiment, and my experiment is designed to test my hypothesis. And so those are the two big important things to take away. All right, so let's talk about the different types of tests that there are, or investigations, we should say. So what kind of investigations in science are there? There's several types. And the first type is about the easiest you can get along with is a descriptive investigation. And so that's where you don't have to do an experiment. You don't have to manipulate anything. You basically bring your test equipment, your recording equipment or your measuring equipment to the site or location that you want to test, or you could bring the stuff that you're testing to you. But either way, you're not manipulating them as much as you can. And so observations are used to answer a question. So I give you an example here. You could say, well, how much does it rain in Phoenix during the month of July on average. And so you would collect rainfall during the entire month of July for 10 years maybe, and then you would average them together and say, okay, this is how much rain it rains in Phoenix. You're not manipulating the rain in any way. You're not manipulating the weather in any way. You're just simply taking a measurement. You could also say, okay, what's the average height of students in this class? You go out and you would take some measurements. You're not stretching the students out. You're not compressing them. You're not influencing their heights at all in any way. You're simply just taking a measurement of their heights. And so descriptive investigations are designed to answer a question about where it's used to answer a question that requires a measurement to be taken. And then the next type of investigation would be a comparative investigation. So, and we'll go into detail on each one of these in just a second. But a comparative investigation is where you're going to control, or you're going to compare how a variable interacts between two or more groups or places. And so let me give you an example of this. So the variable I want to test is what temperature water boils. What I want to compare is at what temperature, or at how does altitude affect at what temperature water boils? And so I could set up an experiment. I don't have a control or a placebo group. I do have an experimental design where I set up a pot of water to boil at 0 sea level, one at 1,000 feet, another one at 2,000 feet. And then I have a thermometer in each one of the pots, and I can look and see at what temperature does that water boil. Because remember, when water boils, that's the hottest it can get at that altitude. And what I might find is that water boils at lower temperatures at higher altitude because there's less air pressure. And so I would graph the relationship of those. But there's no way to have a control group. There's no way to have a placebo group. And so I'm comparing the effect of a variable, that would be altitude, and its effect on the temperature at which water boils. And so I can control how high I am, but I can't control what temperature it boils at. And so that would be my comparative investigation. And here's one for all the stoners in the class: is how is THC potency in just one genetic strain? So that would be a bunch of clones of marijuana. How would the potency vary depending on the soil conditions it's exposed to? Do I just use pure coco coir? Do I use Miracle-Gro? Do I use just regular garden soil? And how do those soil conditions impact the potency of the flower that's produced by that strain of marijuana, or that genetic strain? So all identical. And so what I'm controlling there, I don't have a control, but what I'm manipulating there is I'm manipulating the soil conditions. Just like I was manipulating altitude, I was manipulating soil conditions to see what the impact was on the potency of the plant produced. And then, of course, there's the one that everyone's familiar with, which is experimental investigation. And so that's where all but one variable is controlled, usually just between two groups, sometimes more, to see that variable's effect on the output of the experiment. And the measured output of the experiment could be many things. It could be blood pressure. It could be the ability to be infected by a virus, such as like a vaccine trial. And so what I could say is what is the effect of this blood pressure medication on people's blood pressure? And in this particular case, you would definitely need to have a control group or a placebo group, one that receives something that looks just like the medication but is completely inert. Let's take a look in how these are applied in a more detailed way. What kind of phenomena would warrant a descriptive investigation? So, we know that these phenomena would be something that requires a measurement to be taken, but with little to no interference from the researcher. So like measuring the height of mountains, measuring the heights of people, measuring weights, measuring sizes, measuring rainfalls, measuring temperatures. Those things are descriptive investigations because you don't have to interfere. You just have to take a measurement. So what is the average rainfall in Phoenix? What is the oldest living thing? What is the average speed of cars on the Autobahn? So there's of examples. And so what kind of components do you need for that investigation, for a descriptive one? Well, you need an observed phenomena. I notice it doesn't rain very often in Phoenix, so it begs the question, well, what is the average rainfall in Phoenix? Right? That would be my research question. What is the average rainfall in Phoenix? And then my measured variable would be rainfall in Phoenix. There might be some controls. So I might say, what is the average rainfall in Phoenix during the second week of July. So I might control for the day of week. I might say, what is the average rainfall in Phoenix when the temperature is over 100 degrees? So I might control for temperature. Season, what is the average rainfall during the summer months in Phoenix? I would present my findings either in data graphs, and I would do some sort of analysis on it and draw a conclusion from that. Here I show an example of where a researcher has collected a large number of different types of beetles, and they all have different horn sizes on them, horn shapes. And so you could maybe make some claims about which ones are more aggressive or something like that. But you would say maybe aggression is related to the size of the horns produced. And so you could do a descriptive investigation just watching, observing, recording, and measuring the size of their horns. Okay, so when we get into comparative investigations, let's talk about what kind of phenomena would warrant that. So if we want to take a variable such as like THC potency or at what altitude water boils, we're comparing that variable between two or more groups with no control or placebo, and that's the important part. A comparative investigation appears a lot like an experimental one. The big red flag, green flag, or whatever you want to call it, is that a comparative investigation will not have a control. There won't be a placebo group involved. And so examples, and this is kind of like a foot stomp, like knock knock here. You will see this on a quiz. What type of ant would win in a fight, black or red? That would be a comparative investigation. So if I see a bunch of ants, like a bunch of red and black ants fighting, and I notice that one ant is bigger but losing, I might say like, if it was a fair fight, would he still win? So I could set up a battle royale and test that hypothesis. And then, what's the best type of soil to use? And then here's another example, especially if you're into exercise science, you could say, okay, what type of chest exercise would yield the largest strength gains in the shortest amount of time? So it's another comparative one. You wouldn't have a control group where somebody just pretends to do chest exercises or something, but you would be comparing different chest exercises and then comparing their effect on strength gains among people of similar type, basically. So you wouldn't want to have like, a very small, tiny, like 6-year-old doing this experiment or doing this comparative investigation with, like a bodybuilder. You would want to have like, 30 bodybuilders and then test them and say, okay, which chest exercise among them yields the largest strength gains. So that's how you would control certain variables and their impact on your output. So again, what type of diet promotes longevity? So you'd have to go and see what people were eating and then how that correlates to length of life. And, obviously you would need a large sample size to get past all the noise, because people die for a various number of reasons and not all of them are related to diet. But if you sample enough people, you might be able to draw correlations between what they're eating and how long they live. So here's some of the things that you would need to have for a comparative investigation. You would just need to have something that doesn't make sense that you see, you ask a question about it, and then you create a hypothesis. Okay, I hypothesize that push-ups yields the greatest strength gains because it uses body weight, and the body is best adapted via evolution to deal with body weight exercises. And so I think body weight exercises are going to yield the greatest strength gains. And basically your hypothesis is body weight exercises, because they have an evolutionary history, are going to be more functional than other types of weighted exercises. And then you would test that and then have a prediction that from your experiment, your prediction would say, okay, the people that do body weight exercises will have the greatest strength gains. And so the greatest strength gains is your prediction from your hypothesis. Obviously, you would need your experimental design that would give you your prediction. And then you would need to identify your variables. So what are you controlling for? Perhaps the age of the participants in your study. You would want to keep that the same. Maybe the body fat composition of them. Maybe what time of day the exercises are performed, what temperature they're performed at, and how all of those variables can be controlled and kept the same between groups. And the only thing that you change, the only independent variable you change, is the type of chest exercise that they're doing. And then the dependent variable you would identify as being the strength gains, like what the amount of strength gained after doing those different exercises. And then you would take your data and put it into graphs or tables and provide an analysis of it, and then to draw a conclusion and either reject your hypothesis or accept it. And then you would put forward your recommendations on research. You would say, okay, I found that body weight exercises were not the most effective. I found that this other one was. And so we need to investigate why that one was the most effective. And then you may even present another hypothesis at the end of your conclusion and say, hey, you know what, here's something that could be tested. And so that's essentially what science is doing. And now let's talk about the final one, which is your experimental investigation. And so pay no attention to this picture. It's just meant to show you that you can have this experimental design where it's, complicated, where you're testing one variable that would be your sample, in your designed experiment. And, this is obviously like a physics experiment and not a biology one. And so what sort of phenomenon would warrant these experimental investigations? Well, when you want to measure the effect of one variable on a group of similar things. So that would be something like people who have, what do you call high blood pressure? It's like hypertension. So people over the age of 50 who have hypertension and have diabetes type 2. Yeah, I want to test the effect of my blood pressure medication on that group of people. And that group of people are similar enough that I can, if I split them into two groups and give one a placebo and give the other one my blood pressure medication, I can measure the change in their blood pressure over weeks of treatment and see if there is a difference caused by this one variable. So you're basically establishing a causative relationship between your medication and the outcome of the experiment. And so what sort of things you need for an experimental investigation? You're going to need to see something that doesn't quite make sense, that's of interest to science, that's repeatable. Propose a question like, how did that happen, or how does it affect blood pressure? You come up with your hypothesis. It might be a model for how that particular compound works in the body. And then your experimental design for how you would test whether that compound has the desired effect in the body. The procedure would then give you your prediction. You could say, I think the outcome of my experiment will look like this if my hypothesis is true. Here are my variables. Maybe it's the age of the participants. Maybe it's their health history. And importantly, let's break these down. So an independent variable is any variable that could have a causative effect on the measured output of the experiment. The measured output of the experiment, spoiler alert, is the dependent variable. So you can think about this. Independent variables you can manipulate. You can change them. You can change what time of day that you conduct the experiment at. You can change how much light the subjects are exposed to. You can change the humidity in the room. You can change where you're doing the study at. You can change how much medication is being administered. All of these things are things you can actually change. The tricky part is that you want to isolate just one of those things and keep everything else the same. So the manipulated variable is the one that you want to change to see how its effect is on the subjects in your experiment. The manipulated variable is the independent variable that is changed throughout the test to measure its effect on the dependent variable. And so for your blood pressure medication, the manipulated variable could be your pharmacological compound and how much or what quantity of that compound you're administering to your subjects. So you could have a group that's getting 10 milligrams, a group that's getting 20 milligrams, a group that's getting 50 milligrams, and a group that's getting 100 milligrams, and a group that's getting a placebo. All other controls are all other independent variables like time of day, temperature, humidity, location, body weight, body fat content, whether they have diabetes, their pre-hypertension. All of those variables are kept the same within every group, and so only changing the amount of medication that each group is receiving to include even the placebo group. And so those are what we call control. Think about independent variables as being they can be one of two different things. They can either be a control, something that's kept the same across each experimental group, or the one thing that's changed, the manipulated variable. And that's the relationship of those two things to the independent variable. Finally, you have the control group. So the control group exists as simply a control in the sense that they receive everything except for the treatment. So they have all of the control independent variables like the time of day, the body weight, composition, those sorts of things. And they even receive a pill. It just happens to be an inert pill. It doesn't have any of the medication or the manipulated variable in it. And so that group tells you whether or not there's an effect of just the experiment itself on the output of the experiment. So is the design of an experiment when you get people together in a group and all give them a pill and say we're testing to see if this pill lowers blood pressure, does just being exposed to that situation have an effect on their blood pressure? You've removed the fact that they're receiving medication, and now you're just seeing if the experimental itself, the design of the experiment itself, has an effect on the blood pressure. And so that's what the control group's for. And then you have, of course, the other variable, which is the dependent, and that's the measured output of the experiment. That's the star-bellied sneetch coming out the other side, is that you cannot directly affect the dependent variable. The only thing you can do to affect the dependent variable is to change one of the independent variables. Change the amount of blood pressure medication they're receiving, change the amount of sunlight they receive a day, change the amount of vitamins they take. You can then affect the blood pressure they have, but you can't just like go into their arteries and like, you know, manually squish their arteries or stretch them and make them change their blood pressure. No, you have to do something else. You have to manipulate something that has a causative effect on their blood pressure, but you cannot physically yourself alter their blood pressure directly. So that's the difference between independent and dependent. And typically independent variables are expressed on the x-axis while the dependent variables are expressed on the y-axis of a graph. And speaking of graphs and analysis, you would also do that for your experiment. You'd present your data, come up with a conclusion, and then of course everything else I said. You might go back and say, hey, I know there was this bump in our methods, like a bump in the road, like it kind of, what would be the word? It kind of threw us off track a little bit. And so you may, in your conclusion, give suggestions to future research that may be just trying to replicate your experiment and your results. You might say, hey, change this one thing in your methods, and you probably get better, you get more precise or more accurate results from your experiment. And so that's some things you can do in your conclusion, or you can reject your hypothesis, you can accept your hypothesis, you can present recommendations for future experiments that may help to support your hypothesis or help to support an alternative hypothesis. So there's lots of stuff you can do at the end of your study to help promote and maybe spark some interest in other people reading your paper to pursue a similar subject area. All right, what I want to show you here is this is just an example of a pharmaceutical company breaking down a group of similar subjects, breaking them down into two randomized groups, and then talking about how these two randomized groups are receiving treatment or receiving a placebo. This is literally from a paper that was done by a pharmaceutical company on what is that called? The fluticasone propionate. I don't know how to say that word. It's a word they made up for this study to name their medication, but it has something to do with like COPD, helping people breathe, some sort of oral steroid that's administered to the lungs to help people breathe better. And so what they did was they took 990 people, and these are 990 people that had COPD, I'm guessing, or some sort of respiratory condition that, caused them to be short of breath. So they took 990 of these people and they just kind of split them into two groups. Now they removed some people from the group. As you can see over here, there was some patients withdrawn before. Let me see if I can get my pointer. That helps a little bit. So you see over here, they started with 990, but they pulled 239 people out before they even started this study. So they literally pulled a quarter of the people out that didn't even participate in the study. They got patients and they were like, yeah, let's do it. And then they had to take a quarter of them away before they even split them into groups. So then they randomize people. They say, okay, you guys go in the treatment group, you guys go in the placebo. they gave some tests to them. They gave them some prednisone. The ones that received prednisone, some of them had to withdraw because they had a reaction to prednisone, which is kind of crazy. And then so the ones that didn't have a reaction to prednisone ended up getting the treatment, and the ones that didn't react to prednisone in the placebo group, they received the placebo. And then they did this double blind phase. So what the double blind means is that even the researchers administering the medication itself didn't know whether they were administering the medicine or administering the placebo. It was one group, one level in the hierarchy of researchers above them that knew who was getting what. And so that's why they call it a double blind, is the patients don't know what they're getting and the people administering the medicine to the patients don't know. That's the double blind. And so what that does is that removes any effect of, like, if the researcher is excited about, like, ooh, I've got the medicine, I can't wait to give it to this guy to see what happens. That sort of excitement might transfer to the patient himself, and they might actually, just from that interaction, not from the medication, lower their blood pressure. And they want to remove the possibility of that. And so the researchers administering the medication or the placebo have no idea what they're giving out. And now they do this study over the course of several years. So they do it after 1 year. Only 80% of the people completed the actual program after a year. After 2 years, only 68% were still participating. And then after, what was that? Who knows how long the study was, maybe 4 years, 3 years. Only a little more than half finished the study that was receiving the treatment, and a little less than half finished the study that was receiving the placebo. And then 195 people withdrew. Over half of the people receiving the placebo treatment had to withdraw from the study. Almost half of the people that were receiving the treatment had to withdraw. And so just from the people that finished the study, right, they were able to draw their conclusions about whether or not that actual medication was effective. So it shows you that there's a lot of, like, nuance in science itself, meaning, like, you start with 990 people, you into two groups of 375, and then when you're done, like, you end up with only like 212 and 175 people finishing the study in each group. So that's pretty crazy if you think about it. And it's hard not to ignore the ones that had to be removed from the study and say, okay, why were they removed? I mean, they give the reasons over on the sides, but it's almost like, they could have been removed because of adverse reactions to the medications, the adverse reactions to the sugar in the sugar pill. You just don't know. And so even though this study is done and it was peer reviewed and it's published in a scientific paper, you're going to have to do this again. You're going to have to, that's why they have more than one clinical trial, it's that so much stuff can go wrong during a clinical trial, for a vaccine for coronavirus or something like that. So much can go wrong during the clinical trial that even if they get a positive result, they still have to kind of take it with a grain of salt and say, okay, this is a good outcome. We're happy we got this, but we definitely need to check our work and make sure that this happens every time that we administer the study, every time we do this sort of thing. And so they'll go through phases or clinical trials to try to get the same result over and over again, because that suggests that their medication or their treatment or their vaccine is effective. And so that's why, just to tangent for a bit, because we're kind of finishing up this, right? Just to tangent for a bit, that's why some people were concerned about the vaccine trials with Moderna and Pfizer and stuff, was that they had been on a fast track to approval where had, like one clinical run that looked good, really good. They skipped the other clinical runs to substantiate those results. And so that's why some people were like, well, it seems like we're kind of rushing this thing. But other people are like, well, it works. We should at least like try it and try to get it out there. And so far, it seems like that actually worked out in favor of fast tracking. So that was good. Let's move on. So just to summarize, and then we'll wrap this thing up, is that observations of repeatable, controlled phenomena will lead to questions. Of course, I see something, I ask a question. Questions beg for explanations, and those explanations are hypotheses. We use scientific investigations to test hypotheses. Investigations can be descriptive, comparative, or experimental, and the investigation, once it's set up, can give us a prediction about what we should expect to see from our experiment based on our hypothesis, whether if it's true or not. And then there's a secret thing that says the best way to tell whether it's comparative or experimental is whether there was a test group that was compared to another test group that would be comparative, or whether the test group was compared to a control group or a placebo group that would be experimental. And so those are the big points to walk away from this with. And so I'm just going to exit here. Let's take a look at the note sheet and double check and make sure we got everything. Yep. It sure looks like it. All right, great. Hope you enjoyed this. I will see you guys for the next one.